محمد قاسم انا كان ينزل يروح عنده ولا راح يطلع والله صار هيك 10 ليش مش عندي
انا لا ما انا شنو لا 9 يمكن 9
قاعد 10
سيد سوي 10 سيد سوي 10 ليك شنو وقف سيد تاج راسك تاج راسك
تخلص
تاج راسك يا سام هذا بدون لا يدي عفاته
لا لا والله قاعد لا تجي تصور
from the fact that the mid-sagittal section always shows the whole lumen of the brain vesicle. Other sagittal sections, for example, parasagittal section, will not demonstrate the whole lumen of the brain vesicle. And so, because you are looking to the whole lumen, this is a mid-sagittal section. The brain vesicles are 5 in number in the mature state. It is formed of the most anterior, which is this one, that is called telencephalon. The cavity of it will form the lateral ventricle. Next to it, till here, this is the telencephalon. Next to it, this is the diencephalon, on that side and that side. That will form the thalamus, epithalamus, hypothalamus, and down. And the cavity of it will form the third ventricle. Just look to the formation of the primordium of the epithalamus. This is the primordium of the epithalamus that will form the pineal gland. Next to the telencephalon and then diencephalon, this is the midbrain, which is the mesencephalon. You can see that the midbrain is angulated. And this angulation is called the cephalic flexure. Cavity of the midbrain will form the aqueduct of midbrain, which is called aqueduct of Celia. Next to the midbrain, you can see another region, which is called rhombencephalic isthmus. This narrow region is called rhombencephalic isthmus. It is located between the mesencephalon and the metencephalon, either dorsally or ventrally. So this is the metencephalon dorsally, and that is the metencephalon ventrally or anteriorly. The metencephalon anteriorly will form the primordium of the palm, while the metencephalon posteriorly will form the primordium of the cerebellum. Next to the metencephalon, this is the myencephalon. And you can see that the ventral wall of the myencephalon shows curved-like wall, which are these curvatures are called neuromeres or rhombomeres. While dorsally, the myencephalon is formed by a thin layer of membrane. The cavity of the myelin and metencephalon will be the fourth ventricle. The angle between the myencephalon and metencephalon, which is this one, is called the pontine flexure. The angle between the myencephalon and the motor spinal cord This is the spinal cord. This angle between the myelencephalon and the spinal cord is called the cervical plane. Of course, I didn't consider that section in the description. Why? Because this is not a mid-sagittal section. You can see that not the whole cavity of the brain vesicles is seen. Caudally, you may look to the neural tube that remains cylindrical and it will form the spinal cord. The cavity of this cylindrical spinal cord will be the central canal of the spinal cord. Just look to the dermomyotome and sclerotome on that side and that side. This is the dermomyotome and sclerotome as it appears in the sagittal section. The embryo, during the preparation, is folded due to shrinkage by fixity. And so you can see that this spinal cord is not seen yet at this region because the embryo is folded and the section after the folding will not show the spinal cord. But I can see a mass of tissue around there. This is the notochord seen in the folded embryo. Just look at the tissue of the notochord. Also, this region of the section, however, this is not a mid-sagittal because you can see that the cavity of the brain vesicle is not seen. But anything just ventral to the neural tissue is a notochord, and that is the notochord of tissue. You can see this is the notochord of tissue in sagittal section because it is next to the neural tissue. The neural epithelium of the brain vesicle will form a layer outside it, which is the mantle zone, and next to it, this is the pink layer, is the marginal zone. The same of these structures could be seen in the same stage of embryo, of chick embryo, but in a transverse section. You know that the embryo is folded just like letter C. So when you will have a transverse section, the section will show the two limbs of the C-shaped embryo. The dorsal limb will show the spinal cord in transverse section, while the ventral limb will show the neural tube. Supposing I will consider that part of the section. Here, the dorsal limb shows the spinal cord, and you can see ventral to the spinal cord is the notochord. Look to the dermomyotome and sclerotome. But if I will pass to the other side, to the cranial part, I will see the brain vesicle, and that is the diencephalon. And both the diencephalon and the spinal cord shows mantle and marginal zone. But this is an earlier stage for the mantle and marginal zone. I will show you a more developed stage for these two zones, the marginal and the mantle zone, and in pig, not chick embryo. This is the dorsal limb of the U-shaped embryo, the dorsal limb of the U-shaped embryo showing the spinal cord. The cavity is the cavity of the central canal of the spinal cord. The pseudostructural epithelium is the neural epithelium that will proliferate by mitosis to form the mantle zone. The mantle zone shows two bilateral basal plates and two bilateral alar plates. Neuroblast of the mantle zone will form axons and dendrites. These processes will form this pink layer outside the mantle zone, which is the marginal zone. And outside to the marginal zone, just look to this. Try always to differentiate this as the dorsal root ganglion. It is not the basal plate of mantle zone. It is lateral to the basal plate of mantle zone. So it is a dorsal root ganglion, which is derived from the neural brain. Again, in this section, this is the neuroepithelium. This is the basal plate of mantle zone, alar plate of mantle zone, and the pink color is the marginal zone. This is a dorsal root ganglion on that side and dorsal root ganglion on that side. Just look to the dermomyoclone and sclerotome. I will pass to the other side of the section, and again, I will see the diencephalon because the other side shows the ventral limb of the U-shaped embryo. And also I have a neuroepithelium, mantle zone, and the pink marginal zone. The same will be seen whether in the spinal cord or in the neural tube. And later on, we will see more and more of these sections and more mature status of the mantle and marginal zone.